Hello, my name is Soledad Pesh, and I am going to explain my poster titled Collagen from Haemulon Plumierii Residues, obtained by 3 extraction methods. The co-authors are Sarah Herrera, Gabriela Mela, Juan Carlos Cuevas, and Neith Pacheco. I will explain the abstract. Collagen is a fundamental structural protein in many organisms, including humans, and it is an integral part of tissues such as the skin, bones, tendons, and cartilage. Its importance in materials development lies in its unique properties, such as strength and flexibility, which make it ideal for various applications. Collagen can be obtained from a variety of sources, including fish waste. Worldwide, nearly 70 million tons of processed fish are produced annually, producing 65% waste. These residues can be used to obtain materials of interest, such as skin collagen, bones, and scales. In the Yucatan Peninsula, the Haemulon Plumierii, Coro Coro, chac-chí, ronco arará or ronco margariteño, is an essential species because, although of low commercial value, it is caught in large quantities. Its meat is highly valued and is considered a good quality fish for human consumption. Collagen can be obtained by different extraction methods, such as conventional or chemical extraction, CE, lactic acid fermentation, LAF, or enzymatic extraction, E. This work compared the 3 above-mentioned extraction methods from Haemulon Plumierii skin. In addition, protein profiling was performed to identify collagen in the samples. The yields for the 3 extraction methods, CE, LAF, and E were 0, 1 plus or minus 0, 030, 47 plus or minus 0, 06 and 0, 88 plus or minus 0, 01 respectively. In protein identification, the collagen sample from E showed the characteristic patterns of gamma, beta, alpha-1, alpha-2, and 3 chains, with lower quality the samples from CE and LAF. Thus, the collagen obtained from EE can be used to develop biomaterials for medical or food applications. Nearly 70 million tons of processed fish are produced annually worldwide, generating waste that accounts for approximately 65% of the product. However, these byproducts hold the potential to revolutionize the field of marine biology and waste management. They can be utilized to obtain valuable materials such as collagen from skin, bones, and scales. Collagen, a fibrillar protein that constitutes the connective tissues of the human body, primarily skin, joints, and bones, is among the most abundant in many living organisms due to its structural role in biological frameworks. Type 1 collagen, comprising 25 to 30% of the proteins in the human body, plays a crucial role in maintaining the cohesion of connective tissues. This biopolymer can be extracted from various sources, including marine life. Investigating alternative collagen sources is not just a scientific endeavor, but a potential game-changer in the field, considering that collagen properties differ among species, and that the utilization of waste is a key component of the circular economy. Haemulon Plumierii, commonly known as white grunt, is one of the most abundant fish species in the Caribbean reef areas and is a significant resource in artisanal fisheries in the region. Its capture is often associated with the scale fishing of other target species, such as grouper and snapper. Moreover, it plays a vital role in recreational fishing, making it an essential species for small-scale and subsistence fishing in coastal communities. Therefore, the byproducts of this fish can be used through various methods for collagen extraction. In line with the points above, the objective of this project is to utilize the skin waste of Haemulon Plumierii fish for collagen extraction, testing 3 different extraction methods to achieve this goal. Next, I will explain the methodology. The skin of Haemulon Plumierii was manually removed and then crushed in an Oster-brand blender. The conventional chemical extraction was carried out in 6 steps under the following conditions: deproteinization, degreasing, collagen extraction with acetic acid, precipitation, centrifugation, and freeze-drying. Between each step, washes were performed with distilled water. Regarding lactic acid fermentation, it was performed in 4 steps: an acid hydrolysis with Bromelia penguin pulp extract for deproteinization of the fish residues. Next, fermentation was done with the Enterococcus faecium bacteria extracted from the fruit Prunus salicifolia or capulin. Between each step, washes were performed with distilled water. Subsequently, it was centrifuged and freeze-dried. Finally, an enzymatic extraction was performed, with a pretreatment using 1 normal sodium hydroxide. Then, collagen isolation was carried out using enzymatic hydrolysis with pepsin and 0,5 acetic acid. Between each step, washes were performed with distilled water. Afterward, centrifugation and dialysis processes were conducted for collagen purification. For the determination of the protein profile, the following steps were taken. 1. Each sample was identified. 2. An aliquot was taken from each and dissolved. 3. From the sample dilutions, a volume was taken and placed in an Eppendorf tube. 4. 10 microliters of sample buffer were added to the Eppendorf tubes, and they were processed for proteins. 5. A 10% SDS page gel was prepared. 6. Each well was loaded with a volume of 10 microliters of the samples in the same numerical order. 7. The proteins were separated at 80 to 100 volts. 8. Staining was done for 40 minutes with Kumasi blue, followed by a weekend in destaining solution. Now I will talk to you about the results. In the table, we can observe the yield of each of the extraction methods. We can see that the method with the lowest yield was conventional chemical extraction, where nothing was observed for the protein profile. As for fermentative extraction, the yield was 0.47%, and some faint bands were observed at approximately 36, 18, and 10 kilodaltons. Meanwhile, for enzymatic extraction, the yield was 0.88%, and the collagen pattern of the fish was observed in the protein profile. In the protein profile, when looking at the sample's protein pattern, the E sample remains in its collagen condition as a protein, as the characteristic patterns of the gamma, beta, alpha-1, alpha-2, and alpha-3 chains are observed. Finally, in the graph, we observe how the pH decreased, and colony-forming unit, CFU, growth occurred during the fermentative extraction method. It was applied under control conditions, and using Bromelia penguin extract for the deproteinization stage. Finally, here are the conclusions. The LAF and E extraction methods obtained higher yields. According to the protein profile, E was the best method for extracting collagen from the skin. It is worth mentioning that, in the case of the fermentation method, the Enterococcus faecium bacteria developed favorably during the fish skin fermentation process. The data obtained showed that the treatment with pineapple decreases the growth of Enterococcus faecium bacteria. I would like to thank Pranay 321295 Development and Implementation of Sustainable Methodologies for the Use of Algae Biomass, Fishery and Aquaculture Waste from the Yucatan Peninsula, for its valorization as nutritional food ingredients and functional products, 2022 to 2001 National Research and Advocacy Projects for Food Sovereignty of the FUNHCT. Thank you very much. You can contact me at the following email addresses. I hope you like this work.